Hello, everyone, and welcome to today's webinar titled Novel Engineered Polymerases for the Detection of Infectious Diseases. This webinar is part of the 10th Annual Microbiology Virtual Event Series brought to you by Labroots and is sponsored by Medix Biochemica. For more information on our sponsor, visit medixbiochemica.com. I am Israel Gonzalez of Labroots, and I'll be your moderator for today's webinar. Before we begin, I'd like to remind everyone that this event is interactive, and we encourage you to participate by communicating with other attendees and peers using the group chat found on your screen. If you have any trouble seeing or hearing the presentation, please click on the Help button located in the navigation bar in the event space. Now let's get started. I'd like to present today's speaker, Dr. Maja Studencka-Turski, Scientific Lead at Medix Biochemica MDX. For a complete biography on today's speaker, please visit the presenter window on your screen. Maja, you may now begin your presentation.
Webinar entitled Novel Engineered Polymerases for Detection of Infectious Diseases. My name is Maja Studencka-Turski, and I'm Scientific Lead in R&D department at the German side of the company. I hope you will enjoy the presentation, as the aim is to provide you with some solutions for diagnostic in your laboratories. Before I start, I would like to introduce to you our company. Medix Biochemica is a truly global company. We have our sites in United States, in Europe, and in China, Shanghai. We are providing raw materials for in vitro diagnostics, and each of the sites has its own center of expertise. In Germany, our center of expertise is to engineer polymerases for molecular diagnostics. So we provide you with unique polymerases for DNA and RNA amplification, and the manufacturing and developing of polymerases is taking place according to ISO 13485 standards. We also provide services for customized enzymes development and lyophilization. In fact, many of our products are already available in lyo-ready format, which means you can lyophilize them on site by yourself without the need of adding additional excipients, or we can lyophilize the products for you. So let me first start with explaining you why do we need to engineer polymerases in biotechnology. So as you know, DNA polymerases play a central role in modern molecular biology, and they are necessary in very basic techniques like DNA cloning or DNA sequencing or just simple PCR reaction, but they can be also used in more sophisticated techniques like NGS or synthetic biology. However, if we isolate polymerases from bacteria, from thermostable bacteria, these natural molecular machines differ in terms of their characteristic. They have different degree of thermostability, also extension rate, fidelity, processivity, or specificity. Some of them have this nuclease activity domain. Some of them are able to bypass damage, and the others have, for example, strand displacement activity. The problem with this wild type or natural polymerases, if you will, is that very often they do not have these performance specifications that we need for transformative technologies. And this is where we as a company step in. We can address any of these characteristics of polymerase and improve it so that it will be suitable for an assay of interest, or we can address many of these features and prepare an enzyme which can solve a wide range of problems in the diagnostics. In our company, we engineer polymerases using two different methods. Either we are utilizing a rational design approach, where we required sufficient information about the enzyme in order to do that. Ideally, we have, for example, a crystal structure of the enzyme, so then we can identify, let's say, amino acids which are responsible for processivity or interaction with DNA, and then we can mutate particular enzymes or particular amino acids to gain the function of interest. The other option is to tailor DNA polymerases via random mutagenesis, and this is the approach we are using mostly if we have to develop enzyme for particular assay. So then we start with a wild type enzyme, and this wild type enzyme is then subjected to a random mutagenesis, where we build a library of mutants And this library of mutants is then screened with the assay of interest so that we can identify an enzyme which is performing the best in the particular assay. In this way, we also engineer enzymes for infectious disease applications. However, these enzymes must fulfill following requirements. They absolutely have very high sensitivity and specificity. So we are looking for enzyme which will give us very low limit of detection. At the same time, we do not observe any false positive results with these enzymes. The enzymes must be very processive because in the diagnostic of infectious diseases, the time really matters. Ideally, they also work with crude samples. So this is, again, the possibility how you can save time in diagnostic if you don't have to isolate DNA or RNA from samples. The enzyme should ideally work in a multiplexing setup, so you're able to detect many targets simultaneously, or you are able to detect emerging or mutating infectious agents with difficult sequences. And lastly, the enzymes should be stable at room temperature or should function effectively in a wider range of conditions. As I mentioned at the beginning, many of our products are available in a lyophilized form. So if you lyophilize enzyme, it can be then transported and stored at room temperature, which makes it also usable in testing in low resource settings in the field, or makes it applicable in point of care testing. At this point, I would like to introduce to you some products from our portfolio, which we recommend for diagnostic of infectious diseases. So at first, I would like to start with PlexTag 5x qPCR multiplex master mix. This is our all-in-one solution. It contains engineered polymerase. This polymerase is hot started using aptamer technology. And the entire mix is 5 times concentrated, which means you have a lot of free volume for primers and probes, but also for samples. And this is of particular relevance as PlexTag is working very nicely with crude samples. So it's compatible with blood, with saliva, but also with food lysate matrices. On the top of that, PlexTag was developed in order to work in multiplex setups. So you can use it in real-time PCR multiplexing, but also in endpoint PCR multiplexing with up to 30 targets. And the master mix is formulated in such a form that you can use it also, you can lyophilize it right away. It's lyo-ready. So either you lyophilize it yourself without addition of other components, or we can lyophilize it for you. So this is an example of a simple workflow, how PlexTag can be lyophilized for your assay of interest. So the only thing you have to do upon receiving the product, you have to add oligos and probes into the mix. Then you aliquot it, you lyophilize it, and then you can use this either lyo cakes or lyo bits after lyophilization into your assay. It is important to notice that after lyophilization, PlexTag does not lose its sensitivity, so it performs as good as before lyophilization. So we recommend PlexTag for diagnostic of infectious diseases because it is compatible with high concentrations of blood. So in a traditional approach, using traditional products, one would have to take a blood sample and then subject it to DNA extraction and purification. And then this sample could be used for qPCR analysis. When you're using PlexTag, you can use blood sample directly in the qPCR reaction. So there are no extraction reagents required, no equipment required for DNA extraction. So this is, of course, less error prone, and at the end you get much more starting sample. Just to show you how well PlexTag performs with blood, this is an example of experiment where we compare performance of PlexTag with 3 other competitors using 4 different concentrations of blood. This is starting with 0.1% of blood up to 10% of blood. And we were trying to detect 3 targets, 2 targets from blood. This is coagulation factor 5, actin B, which is a housekeeper, and we also additionally spike the blood with internal control. So as you can see, PlexTag was the only mix which performed consistently with less variability in CQ values with all blood samples. Was the only one which was able to amplify all of these targets in 10% of blood, whereas competitors either fail completely to amplify in this high concentration of blood, or they failed to amplify all of the targets. This is another way of visualizing this data. We load these PCR reactions at the end on the agarose gel to look specifically for the amplifications, and we can see that for 10% of blood, Plextag was able to amplify all the 3 targets of interest, whereas the best performing competitor 1 managed to amplify only 1 of the 3 targets. So seeing that, we decided to challenge further Plextag and test how it performs with higher concentrations of blood. And for that, we use different types of blood samples and performed a comparison between performance in 0.5% of blood and 20% of blood. And as you can see, there is basically no difference in amplifications of the 3 targets depending on the concentration of blood in the sample. So it shows that Plextag is very highly tolerant to inhibitors in blood. In fact, it is also very tolerant to inhibitors in food matrix crude lysates, and as such, it can be used for water or foodborne pathogen screening. And this makes this Plextag also very usable in foodborne screening assays. This is an example of an assay that we developed for our customer who requested Plextag in the form of lyophilized lyo cakes containing already primers and probes for specific bacteria for waterborne or foodborne screening. So these are the examples of performance of Plextag for waterborne screening bacteria and for foodborne screening of pathogenic bacteria. You can see we are able to detect up to 5 targets with Plextag from this difficult food matrix samples. So to summarize on Plextag, this is our widely used master mix, which we recommend for multiplexing assays. It is formulated in such a way that it can be lyophilized right away, or we can provide you with lyophilization of Plextag. It works directly with crude samples, so there is no nucleic acid extraction required. All of these very good features you get for very competitive price. The second product I would like to draw your attention to is our HIDE polymerase. HIDE, in contrast to Plextag, is designed for very specific amplification. HIDE stands for high discrimination. So we recommend this product, this polymerase, for allele-specific PCRs because it shows very high sensitivity. It can detect 10 copies of mutants in a background of over 10,000 copies of wild types. So it has this very excellent discrimination. On the top of that, HIDE is hot started, so it doesn't show false amplifications in the reaction. And it also tolerates inhibitors from blood in up to 5% of whole blood in the final reaction. So it can be used for rapid genotyping or screening. So what makes HIDE so unique? As you probably know, most of polymerases will amplify targets even if there is a single nucleotide mismatch in the binding, in the primer binding. And HIDE will not do that. It will identify this mismatch and will not amplify such a product. We achieve that by our rational design approach, where we identify all the amino acids which are interacting with the phosphate backbone of the primer. And then we mutated the amino acids and looked for the enzyme which gives us the best selectivity. And then we identify HIDE. So this is the example how simple it is to use HIDE in the discrimination between mutated allele and wild-type allele. The example is coming from a well-known mutation in the BRAF gene. BRAF gene is a common driver of cancer diseases. And while using HIDE, you have to only remember that the discrimination is driven by 3' mismatch. So what it means basically for you that you have to design a forward primer with last nucleotide matching the allele of interest. This can be mutated allele and mismatching the wild-type allele. If you design it in this way, you will see how well HIDE discriminates. So HIDE is able only to amplify the matching template and is failing to to amplify mismatching template. When you look on the performance of competitors either to fail completely to amplify the matching template, so they show very low yield, or they show very poor discrimination. So these are the examples how you can use Heidi, or how Heidi was used by our customers in their research. So the first example is coming from a comparison or identification of HIV variants. This study focused on identifying inhibitor-resistant variants of HIV. This inhibition is driven by mutations in VIF protein, and using Heidi, they were able to discriminate between these two variants of HIV. The other interesting study is coming from studies on Neisseria meningitidis. This is a bacteria causing meningitis, and this bacteria may sometimes show a mutation, which gives it ciprofloxacin resistance. And the research group was analyzing perform comparison between data from DNA sequencing and performance with Heidi, and they observe a complete correlation between these results. However, while using Heidi, you get results under 3 hours, so which is of particular relevance if you're screening for antibiotic resistant bacteria. So using Heidi, they were able to get quicker results and to faster apply proper antibiotic therapy. The last example is coming from studies on fungi, in particular on identification of aspergillus types, which are producing mycotoxin. So there are the group was interested in profiling of aspergillus fungi to identify those types which produce STG mycotoxin. And we know that this mycotoxin is present, this type of aspergillus also present in our homes. So they were interested in a reliable discrimination between toxic and non-toxic variants. So to summarize on Heidi, Heidi stands for high discrimination. So this polymerase gives you a superior SNPs detection. So it outperforms competitors in this ability to distinguish a mismatch in a primer binding. It is recommended for rapid screening as it tolerates up to 5% of whole blood. It also, by this rapid screening, it allows for very quick decision, potentially without waiting, for example, for culturing of bacteria to see which one are resistant to antibiotics, or waiting for sequencing results. So we recommend Heidi for applications such as antimicrobial resistance gene screenings or species identification. The next group of products I would like to introduce to you is a product ideal for RNA applications. And this is our Volcano RT-PCR master mix. Volcano stands for a very unique of our engineered polymerase, namely, it's able to simultaneously perform reverse transcription and amplification. And because this reverse transcription takes place at higher temperatures, therefore we call the product Volcano. Volcano is a ready-to-use master mix. It's very simple to use and it's sensitive. It gives you very fast time to results. It contains polymerase, which is hot started using aptamer technology. And importantly, as the reverse transcription takes place at higher temperature, we recommend Volcano for very complex samples with secondary RNA structures, because when you perform reverse transcription at higher temperatures, you do not observe anymore these secondary structures of RNA. So to show you how hot is the reverse transcription with Volcano, this is a comparison analysis of primer extension assay for standard reverse transcription and for Volcano. So standard reverse transcriptase, as expected, does not perform any extension above 50 degrees. However, the optimal temperatures for Volcano are ranging between 60 to 75 degrees. So again, performing reverse transcription at this higher temperature can be beneficial for assays dealing with complex RNA samples, but also you have to keep in mind that at these higher temperatures, no RNA activity is observed. So it gives you also higher specificity of the assay. Another benefit of reverse transcription on higher temperature is that you don't have to perform initial reverse transcription isothermal step and then follow with cycling for DNA amplification. You can combine all of this within one step. So you can use so-called zero-step RT-PCR protocol, where reverse transcription and annealing extension are taking place at the same temperature simultaneously. So this gives you also shorter protocols and saves time to results. So you can use this very simplified protocol on the top of that with a master mix, which is also tolerates crude samples and is compatible with, for example, if you're interested with DNA intercalating agents. So you can combine it, for example, with Cybergreen. This is an interesting example how Volcano can be used in screening for infectious diseases. The example is coming from University of Miami, where they established a screening assay using Volcano as a master mix. The screening assay comes from wastewater, where they screen for SARS, for COVID infections, and they also screened water for monkeypox virus infections. So what they observed, that using Volcano, they could save a lot of time, first of all, because they were able to use extracts from wastewater directly as a sample in the master mix. And secondly, there was no requirement of additional cDNA synthesis step. So they save a lot of time in the qPCR reaction. On the top of that, they also established an assay for monkeypox screening, where they could combine screening for both RNA and the DNA templates. So this is the advantage of Volcano. The polymerase is able to amplify RNA and DNA, and as it utilizes both templates, you can, for example, use Volcano in a duplex screening when one of the targets is coming from a virus, and second, it's, for example, your human contamination control. So these screenings are good examples of detection of infectious diseases, and as both of these diseases are currently emerging again, we find it of particular interest to mention about those two. So again, for our RNA applications, we recommend Volcano mix. This is a master mix which is recommended for RNA, but also DNA target screening. It contains hot-started polymerase. It works with crude samples. It's very simple to use and sensitive. And in addition, it is lyo-ready. So if you wish, you can lyophilize it, but we can also provide you with lyophilized form of Volcano. The last group of products I would like to draw your attention to are our products for isothermal amplification. As you may know, isothermal amplification, when combined in a LAMP reaction, is a very interesting and low-cost alternative for detecting certain infectious diseases. And it's also alternative for very expensive real-time PCR methods. And on the top of that, if it's combined with reverse transcription, then it's also usable for detection of viral infections. So this is a summary of advantages of LAMP in molecular diagnostics. So as you know, for LAMP reactions, you don't need very specific cyclers, very expensive equipment. It gives you results in very fast, usually under 15 minutes. It's very easy to visualize. This is an example of colorimetric LAMP reaction for COVID screening, where all the blue samples are positive and the purple are negative. So it's very simple also to operate. And for that, we recommend it for point of care testing or, for example, at home use. So the first product in this portfolio is our FastBST polymerase. It is warm started, so you can use it in room temperature. We recommend it for isothermal amplification of DNA targets. It's very fast in performance and it can be used in point of care testing. It is lyo-compatible, which means you have to add some excipients required for lyophilization into the mix, but the mix itself can be lyophilized. It is available as a separate component, but also as a master mix. So to show you how fast is this BST polymerase, this is a comparison of the performance of FastBST and some other competitor. And when you look on the detection times, you can see that FastBST is approximately 10 minutes faster than the competitor in every dilution of the DNA sample, also much faster in the lowest concentration of DNA, which is 10 copies per reaction. And in this lowest concentration also gives you more reliable and consistent data. On the top of that, FastBST is also compatible with inhibitors from blood. So when you look on this diagram below, you see that if the reaction was performed in the presence of 0.5% of blood, FastBST was still performing very quickly and much faster than the competitor. In a collaboration with Italian Institute of Technology, we were able to establish point-of-care microbial testing, where FastBST lyobits performed by our company were utilized in this point-of-care testing using crude samples. So this is a simplified workflow of the procedure, where first a crude sample will be collected. It can be saliva, it can be blood. And then this crude sample is then subjected to extraction for 5 minutes, and the extraction solution is then pipetted onto the reconstituted lyobits. So usually the reaction time is between 10 to 20 minutes, and the sensitivity is depending on the assay, but is as low as between 2 to 10 copies per reaction. They were able to identify a lot of very difficult bacteria, for example, like Mycobacterium tuberculosis, which is rather bacterium too difficult to work with. And they were able to see that with FastBST, they could detect as little as 10 copies per reaction of the bacteria. They were also successfully able to identify some other targets in very interesting matrices, like, for example, biosolids or in milk or nasal swab. The other product in the isotherm amplification portfolio we recommend is our isotherm 3G. This is an engineered polymerase, which we engineer so that it can work in LAMP and RT-LAMP. So it can amplify DNA templates, but also RNA templates. It is also warm-started as BST. We recommend it for point-of-care testing. In this case, isotherm is available as a separate component and also as a master mix. And it is lyo-ready, which means you can lyophilize it without addition of other excipients. So this is an example how isotherm 3G performs with RNA template. In this case, we analyze amplification of N gene, of SARS-CoV-2 gene, and you see that isotherm is much faster than the other 4 competitors. And in fact, on the top of that, it's not only the fastest one, it's also very sensitive. And what makes it very unique in comparison to the other competitors is that we are not observing this false positive signals in negative controls, which were very commonly observed with others. Isotherm 3G was also tested by Italian Institute of Technology with DNA samples for bacteria screening, and they were able to show that isotherm is very sensitive and performs very fast amplifications. So under 10 minutes, they were able to detect 10 copies of Salmonella or Legionella, and the same results were also observed for Staphylococcus or Pseudomonas. And similarly to FastBST, isotherm 3G is also inhibitor resistance. We tested the polymerase in the presence of 7.5% of blood in a final PCR reaction and with 15% of saliva in the reaction. And as you can see, in all of these crude samples, we observe reliable amplification of both DNA and RNA. So for isotherm application, we have 2 products. Both of them are warm started, so they can be set up at room temperature. They can be used for point-of-care testing. They are very fast, and they are available as a separate components or as a master mix. FastBST is recommended for very fast DNA detection. It is lyo-compatible, whereas isotherm we recommend for LAMP and RT-LAMP, where you can use it for DNA and for RNA targets, and it is available in lyo-ready format. So at this point, I would like to briefly summarize on our products. as you could see, our portfolio has a range of products with very unique advantages, but what all of them have in common that they are very often warm started or hot started, which makes the setup much easier in the laboratory. They are very often available in a form or in a lyo-ready form, so we can lyophilize for you. So in the case of testing outside of laboratory, this is a good alternative, for example, for point-of-care testing. And many of these products work very nicely with crude samples, and they are also very processive. And this is of particular relevance if you're interested in diagnostic of infectious diseases. So at this point, I would like to thank you for your attention, and I would be happy to answer your questions.
Well, thank you very much, Maja, for that informative presentation. Let's go ahead and dive into the Q&A. Joining us as well as Maja, we have Rob Thompson, Global Product Manager for Medix Biochemica. Now, as a reminder to our audience, you can submit your questions below in the question submission box for Maja and or Rob to answer live. Let's go ahead and dive into it. We do have a few questions off the bat. Let's go ahead and start with this first one here. Is PlexTac compatible with crude samples other than blood?
Oh, Maja, I think you might be muted.
I think she is. Give me just one moment. Let's figure this out here. Well, Rob, I'm going to pass it over to you while we get Maja's microphone reconnected here. Is there anything that you would like to address to our audience?
Sure. So other than blood for PlexTac, we've also done work with saliva, and we're also investigating it not in the infectious disease space for humans, but in kind of the plant space as well, looking at plant samples with blood. And plant samples are always very challenging to work with, lots of inhibition there. So yes, other than blood, we've worked with saliva. We're working with plants at the moment.
See, thank you so much for that. All right, looks like Maja's going to refresh and come back into the studio, so we're just going to wait for her to rejoin. Thank you to our audience for your patience. In a world that we live in right now where we have mostly virtual meetings, we all know that these kinds of technical issues can happen, so appreciate your patience.
Can you hear me now? Sorry.
Yes. Yeah, that works.
There we are.
My apologies. Sorry.
That's okay, Maja. Thank you so much for your patience as well. I'm going to go ahead and ask that first question one more time for you, Maja, if you don't mind, which is PlexTac compatible with crude samples other than blood?
Yes, it is. We tested PlexTac with saliva, but also with plasma samples, and it's also compatible with plant samples.
All right, thank you for that.
Yeah.
Go ahead and move to our second question here. Heidi composition and principle of working. Seems like there's a question about that. Maybe you can talk more about it.
So Heidi, the principle of how Heidi works, it's able to discriminate between the difference in the last nucleotide at the end of a forward primer. So if the forward primer matches, for example, a mutated form like a SNP, let's say, in a template, then it will just detect this one. It won't detect mismatching at the 3' of the forward primer. This is how the Heidi discriminates. This is how it is designed to discriminate. And it can be mismatch between AT or AC or CG. Yeah. And the second part of the question, sorry, could you repeat that? There was a second part, I guess.
Yeah, so Heidi composition and principle of working.
As a composition, this is a we have a Heidi available in a as a Heidi tag, but so it can work. It's compatible with Tagman probes, for example. But we also have a Heidi which can work with hybridization probes. If you're interested in this, yeah. So these are the options we have in our portfolio.
Great, thank you for that, Maja. I'm going to go to this other question here. They say, really, I'm wondering the stability of an RT. Any clarification is appreciated. Stability of an RT.
Then I would need to ask some additional questions. For example, our volcano, if we lyophilize it, then it's stable at room temperature over 2 years. But it's also because it's hot started, it can be also stable at room temperature for quite some time. So yeah, you can leave it, for example, for 1 hour. If for whatever reason you get distracted during your setup, then you can leave a volcano on the bench, do your stuff, and come back, and then it will still work.
Understood. Thank you for that. Move on to another question here. Are your polymerases fast? What is the minimal anneal extension time? What is the total time for 40 PCR cycles?
Of course, it's dependent on the product. If we take, for example, PlexTag as an example, PlexTag has a Tag polymerase inside. So as you probably know, Tag polymerases are able to process around 1 kb in 60 seconds. So, I mean, what we tested was amplification, so annealing extension time of 5 seconds, and it works. However, you have to always think what's the size of the amplicon. So, if the amplicon is small, it's like around 60 base pairs, then it obviously will work. If the amplicon is a bit longer, then we would recommend longer extension times. And the entire time of 40 cycles, it would be around 45 minutes. But again, it depends on the what's the ramping time, if you're using fast protocols, or if the ramping in your device is it's slower, then it will be slower. However, it will be for most of the cases under 1 hour, or around 50 minutes.
Great, thank you for that. We have another question here. How isothermal 3G we use to identify a pathogen?
With isotherm 3G, you can, of course, there is a specific set of primers required recognizing specific pathogen. Let's say the example we are having, it's from virus, it's N-gene. So there is for LAMP amplification, you require the specific set of primers. It's like 6 different primers for amplification, and this is how you can detect specific either pathogen or a virus or bacteria. Yeah.
Great, thank you for that. We have one more question here. which is, could you explain a little more about the engineering part of the presentation? I am interested to know how you choose whether to go for random mutagenesis or the rational design approach.
Yeah, so as I mentioned at the beginning of the talk, if we have enough information available on that enzyme, let's say there is a crystal structure of an enzyme, then we can identify some amino acids which are, let's say, in the enzymatic pocket, or they are in the direct contact with DNA or with nucleotides, and then address these amino acids and mutate them. If this is not the case, we don't have enough information, then we start with random mutagenesis. And this is usually where we start if we have to develop enzyme for particular assay. However, we also mix the two techniques. Let's say we start with random mutagenesis, then we get some candidates, and we feel we can still improve the performance. So on the top of new mutant, we would mutate particular sites where we know, okay, it will increase, let's say, specificity or processivity of the enzyme. So it really depends what is the question, where is the assay for which we have to develop the enzyme. If this is, for example, it's supposed to work with purified DNA, or it's supposed to work with crude samples.
Awesome. Thank you so much, Maja. Now, before we end the session today, Maja, I kind of want to give you the floor one last time. Do you have any final comments to share with our audience today?
Not on the top of my head. I would like to just remind you that many of the enzymes that we are having, they are live ready, and this makes them very easy to use. So we would encourage you either to ask us for help to lyophilize enzymes for you. We can also customize the development and customize lyophilization for you if you're interested. But we were really encouraged to test our enzymes. Thank you.
All right. Well, thank you again, Maja, for your time today and for your important research, and for Rob for joining in this Q&A session. Before we go, I'd like to thank our audience for joining us today and for their interesting questions, and to our sponsor, Medix Biochemica. Questions we didn't have time for and those submitted during the on-demand period will be addressed via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until September 5th, 2025, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. We also encourage you to continue venturing out and exploring the virtual space laid out before you. Check out our poster hall, the social space, and even our exhibit booths, where Medix Biochemica has a booth that you can check out and speak more with people like Rob and Maja. Until next time, goodbye, everybody. Thank you.
Thank you.
Thank you.